Hello everyone, my name is Yixin Li. It's a great honor to have this speech here. Today I'm going to talk about how to reveal coral growth patterns through the three-dimensional reconstructions of canal network. Reef-building corals provide complex 3D niches for various species. However, coral reefs are currently declining globally due to the climate change, especially owing to the intensification of El Nino, water quality deterioration, and overexploitation. Studies on the biological process governing the coral reefs and their ecological health and their environmental changes are therefore in the spotlight. As the heart of healthy reefs is sustained formation of the calcium-carbonate skeletons of many coral species. Therefore, it is important to understand how coral grow their skeletons. The structure, distribution, volume ratio, and other useful patterns of the canals in coral skeletons are also important to understand, as they play key roles in coral growth. Traditional biological analysis techniques such as electron microscopes and grading sections need a high workload and complex operability. They provide limited evidence due to their non-transparent coral skeletons, which restrict the direct observation of the structures and related parameters in coral colonies. In recent years, with the development of popularization of HRCT, researchers on coral skeletons has reviewed the morphological and internal structures of coral branchlets and also investigated coral skeletal structures and exogenous influences on coral skeletons. However, there are various changes in studying coral growth patterns regulations using CT sectional slices and signal dependent on skeleton reconstruction. To fill the current knowledge gaps, we created a novel method of canal reconstruction, which use HRCT to study coral growth pattern regulation in polyspora cornis, visualizing its catalyst network, binding signals, polyp relationships, colony growth direction, and coral branching regulations. The reconstructed images present the complete form of the original coral skeleton details of surface buggles and calyces, which significantly facilitates the study of its skeleton and biological characteristics. To investigate the growth pattern of polyps, we reconstructed the calyces and interceptor spaces chamber by chamber. The growth patterns of polyspora can be recognized as generally three-stage growth structure based on three growth bundles along the dichotomous growth axis and assembled by multiple growth types. As a important aspect of coral growth, we consider a group of corals that divides from the same corals as a growth bundle, which contain all the above described growth types, and the growth direction of the bundles depend on the growth axis direction of its branch. Our work presents an application of cutting-edge technology in investigating reef-building coral structures and reconstructing the internal catalyst network based on a large column of polyspora cornis. We studied the growth patterns through budding growth and distribution information obtained from the reconstruction of coral catalyst network. It reveals that each catalyst of polyspora is a basic polyp growth unit, and growth bundles are arranged along the axis in a dichotomous branching pattern. The growth pattern of polyspora cornis lays the foundation for further explorations on the monthly, seasonal, and yearly growth of repeating corals. Furthermore, we reconstruct the entire canal network in polyspora cornis by HRCT, which sheds light on studying the process of coral formation and budding patterns of living polyps. Our result also suggests that HRCT can be used to characterize the development and process of core growth. Light plays a vital role during the growth of the corals. Different environment conditions affect callus structures within the same colony, and they can be classified into low light and lighted mineralization areas and low light and low lighted growth areas based on the modeling. The measurements in the callus constructions uh, show the apparent differences between light areas and low light areas. To sum up, the three-dimensional morphological reconstructions on the callus and in possible demicolonies reveal a method of studying the biological characteristics and growth patterns of refilling coral species. After that, we expand this method to determine the growth pattern in much more predominant refilling coral genera. Additionally, the basic growth patterns of coral colonies are different to quantify, and calculating skeleton values through HRCT reconstructions can mitigate this issue. In this study, we can reconstruct 11 coral species using HRCT to investigate coral growth patterns and parameters. Our skeleton and canal reconstructions revealed the characteristics of different coral species. We performed a series of experiments, including lumen reconstructions of the canal system in each colony, growth axis formation, growth ring visualization, grayscale gradient maps, and growth parameters quantification to obtain the formation mechanism and related parameters for our colony samples. According to the canal reconstructions and measured growth parameters, we investigated different growth strategies of the major species to define how corals grow their skeletons and how skeletal growth varies among species. The corals of each species have their own uniquely reforming strategies. The canal systems in the skeleton of a colony is the basic foundation for coral growth. All these canals integrate the coral branch into an entirety like the circulatory system in higher animals. We further visualized the growth axis and growth rings to understand the coral growth directions. The type and direction of the growth axis determine the branching pattern and growth direction of the colony, respectively. We drew a skeleton grayscale map and calculated the coral skeleton's void ratios to ascertain the skeletal diversity, devising a method to quantify coral growth. The skeletal density of coral colony is found to be related to seawater temperature, rainfall, and sunshine hours, while the density of skeletons in each growth range fluctuate quasi 12 months periodically. Therefore, the grayscale gradient map can be compared to the growth ring reconstructions to study the monthly and yearly influence of the marine environment on coral skeletons. The related growth parameters can be obtained by reconstructing and measuring the canals in each growth ring along the growth axis and performing cluster analysis to infer the light conditions received by the colony when this part of the skeleton is formed. This research increased the breadth of knowledge on other reef-building corals grow their colonies, providing information on reforming regulations. In the complex canals in coral colony connect all polyps into a holistic network, enabling them to collaborate in performing biological process. There are various types of canals with structures that are dynamically alerted during different coral growth stages due to the internal calcium transport. in this study, we investigated the morphological changes in the correlate of Acropora muricata samples by high-resolution computed tomography, observing the patterns of calcium carbonate deposition within exocorrelite during processes of new branch formation and truncated tip repair. We visualize the formation of a new branch from a calyx and the calcium carbonate deposition in the exocanal. Furthermore, the diameter and volume changes of the exocanal in truncated branches during rebuilding processes were calculated, revealing that the volume ratio of calcareous deposits in the exocanal exhibit significant increases within the first 3 weeks, returning to levels in the in-situ state in the following week. This work demonstrates that calcium carbonate can be stored temporarily and then remobilized as needed for rapid growth. The results of this study shed light on the control of calcium carbonate deposition and growth of the exocorrelate in Acropora. This self-healing process infers that transport within the gastrovascular system is responsible for supplying the needed materials for deposition to occur, and then dissolution of the calyx that removes calcium carbonate and that restores the original diameter of the exocorrelite and exocanal to occur. The removal of calcium carbonate can be used for the linear growth of the skeleton. The data in our work revealed a longer period calcium deposit and release processes during the self-healing process, demonstrating that calcium carbonate can be stored temporarily in calyx and then remobilized as needed for rapid growth. This study expands our understanding of calcium transport patterns in Acropora, which sheds light on the control of calcium carbonate deposition and growth of the exocorrelite in Acropora. Data enable a much more detailed study of the tissue during the build-up of calcium carbonate and conversely during the dissolution of the exocorrelate in the 28-day period. This could contribute greatly to an understanding of the process of calcification. In this study, we reconstructed 11 coral species using HRCT to investigate coral growth patterns and parameters. Our reconstructions of canal network revealed the characteristics of different coral species, and we further visualized the growth axis and growth rings to understand the coral growth directions. Our reconstructions can visualize the coral growth processes during different physiological states, which reveals the mechanism of calcium transport in coral colonies. In addition, we calculated the coral skeleton void ratios to ascertain the skeletal density and devising a method to quantify coral growth. On the basis of the 3D reconstructions and growth parameters, we investigated the growth strategies of different coral species. And this research increased the breadth of knowledge on how reef-building corals grow their colonies, providing information on reef forming regulations. The data obtained through HRCT and its 3D reconstruction contain a large amount of coral growth information, which can be used in further research on reef forming patterns under different conditions. The method used in this study can also be applied to animals and with porous skeletons. Thanks for listening.